All right, I think I will get started. It's very nice to see a lot of familiar face here. Also, I pretty glad I was following Iran. Iran already spent a lot of time to describe the background, so I don't have to do it again. And I would like to thank the organizers give me this opportunity to present our work as well as a web portal we previously built for visualization. The tool or the website I'm talking about today is called Catlas. It's for short for cis element atlas. To show you the function for this web portal, I would like to basically using one of our previous publication as an example. And then later, I will also talk about a few more data sets. If people are not only interested in the brain, we can offer other data set for you to explore. So first of all, I like to give you a little bit background. I will go through this very fast. So I know there's a lot of tools already developed for the single cell RNA-seq data, and there's no doubt that single cell RNA-seq data really open our eyes to identify those new cell types, as well as evaluating the expression patterns for different cell types. So in our group, Dr. Bingren's group in UCSD, we are really interested in understanding the regulatory mechanisms of the gene expression. So that's why we are quite interested to map those cis-regulatory elements, or you may be familiar with some of them called enhancer or promoters. So in the old days, like in the ENCODE and the roadmap project, people use ChIP-seq, different DNA-seq, or also the TAG-seq data to map those cis-reg elements in bulk level. But right now, with the help from Dr. Sebastian Priso and also another research associate in our group, who adapted this combinatory barcoding approach to profile the TAG-seq signals from single cell. So we developed this experimental pipeline and apply it to one kind of the tissue, which is I'm going to talk about today, is the mouse brain. So in the first version of the Samba paper, we mainly focus on half of the mouse brain. So we basically dissect the brain into several slices, and for each slice, we further dissect them into different brain regions based on the annotation from Allen Institute. So roughly, today the data set I'm going to talk about is more than 800,000 nucleus from 45 brain regions. And I think right now we are working on mapping the other half of the brain. We're also hoping to release this data soon and also update those data in our web portal so that you can explore it. So in summary, I guess I will basically go through several major findings and also how you can use our data to ask questions or answer your own questions. So first of all, here is an overview of how many cell types we find from this data set. In total, we identify 43 major types. And for each major type, we further extract them out and perform another round of clustering in order to find the subtypes. So in total, there's in total of 160 subtypes. So for each cell type, we annotate the cluster basically according to the previous published marker genes. For example, let me show you one example. For example, here we annotate one of the cluster as a SST positive GABAergic neuron. Because we find actually the open chromatin signals are quite significant only in this cell type at the SST loci. And as I mentioned before, we extract the cells from for each major type and perform another round of subclustering so that we can find multiple subtypes here. For example, again, the SST GABAergic neuron can further divide it into 10 subtypes. And one of them have this open chromatin signals at the trodo gene loci. That's why we can further annotate this subtype as a trodo cell, or also known as a long projecting GABAergic neuron in the brain. After annotating those data, I will also double check with the annotation from RNA-seq data. So this is the integration analysis. I'm showing you here is a confusion matrix. I'm showing you that our taxonomy identified from a taxic data are very well aligned with previous published RNA-seq data. And you can see some of them actually we can find more subtypes in the taxic. So since we dissect the brain very precisely, we have this regional information. So it's not a surprise we can color each cell based on the brain region information. And you can see some of the cells actually show a gradient difference across different brain regions. Let me show you this slide. Maybe it's more clear to you. For example, here's the bar chart for 160 cell types we find. And for each bar, we color the cell. We basically present how many of the percentage of cells came from different brain regions. And based on this information, we can basically calculate the specificity score to demonstrate whether this cell is more distinct in one particular brain region, or they're more likely to be broadly distributed in the whole mouse brain. So again, here's another example. We find several subtypes for PV positive GABAergic neuron. And when we color the cells based on the brain information, you can clearly see there's a subtype are quite enriched in the striatum area. And we can use the open chromatin signals at the gene body region to find several marker genes from our data. So after we identify those cell types, we can definitely call peaks from the taxic data. And here I show you one heat map mainly to show you that actually the peaks are quite specific to each cell type. Although you find there's one group of candidate-cis element, we call it as a module 1. Those elements are likely to be accessible in all the cell types. That is because majority of those elements are located as a promoter region, or they are overlap with the CT-set binding site. But the whole point is most of the candidate-cis element we find are showing this cell type specificity a lot. So using those candidate-cis element we find, you can really ask several questions. You can simply download the data from our web portal, and also you can also download the results from differential analysis we did previously. So I'm just showing you one example. I mentioned before, this is the SST trodo cell we find as one of the subtypes. And you can identify the differential peaks for this population. And from this population, from the differential peaks for this subtype, you can further do some motiful enrichment analysis and to identify several potential driver TFs. And this is also valuable to check the other modality. For example, you can check the expression level from RNA-seq data so that you can make sure which TF are more likely to be the driver TF. Another thing I want to mention is we are not only providing a list of candidates elements, we also tried to link them to its potential target gene. So our strategy is very simple and straightforward. I think with more and more single cell data released recently, we can definitely make it better. For example, we can incorporate other informations like the chromatin contact from M3C data or the other modalities. So our strategy is like we first calculate the co-accessibility between every peaks. And then if the co-accessibility score is high, that means these two genomic locus are more likely to be close to each other in a 3D space. And then for every pairs we find here, we further examine the correlations between the accessibility signals at those peak regions, as well as the RNA expression for its potential target genes. And if we find a positive correlation between the candidate cis elements with its target gene, we will call this more likely to be a pair, and we will assign those candidate cis elements to its potential target gene. Of course, we set its cutoff against to a random shuffle background. So here is one of the figure I want to show you is two heat map. So the left one is the accessibility signals for every candidate cis element we consider as a potential enhancer. And at the right side is the expression level for its potential target gene. So you can see they're quite well aligned with each other. So this could be another information you can assess from our data. And since we are unbiased sampled the cells from the mouse brain, you can not only examine your interested cell type, you can also ask few questions in several lineages. For example, the oligodendrocyte maturation in the brain. And using the candidate cis element, you can identify the potential driver TFs for every cell type along the maturation of oligodendrocyte. And also, we also captured the adult neurogenesis in the mouse brain. They are mostly located in two brain regions. One is called the subgranular zone, and the other is the subventricular zone. Similarly, you can identify the driver TFs. Here is my last example. I will basically using this example to demonstrate the web proto. So here I have three cell types. Those are the radioglia-like cells from subventricular zone, and the radioglia-like cells from subgranular zone, as well as the potential neuronal intermediate progenitor cells. So here I color this three cluster in red, blue, and purple. And in the lower UMAP, I color the cells based on the brain region. So you can clearly see, it's not surprised the radioglia from subgranular zone are only came from hippocampus. But you can really appreciate that you can see actually there's two populations of neural progenitor cells. Some of the cells actually came from the subgranular zone, and some of the cells came from the subventricular zone. So of course, you can do a differential analysis between these two populations of progenitor cells, identify the potential differential peaks. And as I said, we previously linked those peaks to its potential target genes. So I just show you two examples here. You can see there are several peaks here are more likely to regulate the expression of NeuroG2. And this gene is quite important in the neurogenesis from subgranular zone. As well as another example, there's another enhancer more likely to regulate the expression of TRA-PPC9. This gene is also quite important in the other neurogenesis zone. Okay, so I think I have roughly go through the findings we have, and let me briefly discuss the functions for this web portal. So this web portal is mainly designed to visualize the data and offer people to assess the results we find in this data set. We mainly offer three functions. The first function is called a cell cluster. We use the UCSC cell browser to embed all the cells so that you can explore them in different level. I will show you a demo later. And the second function is called a cell type. And we basically provide us detailed annotation for each cell type, as well as some QC matrix for every cell type. And I think also the browsing the tracks is also very valuable. So we provide a genome browser as a function. Let me now go to a demo. Can you see my web browser?
Yes, we can.
Okay, great. Let me also put this link to the chat. Okay, this is what I'm talking about. We have this web portal, and we offer three functions. So let me go to the first function here called the cell type. So basically, we offer the people to explore the cell type at different level. For example, you can explore the cell type, all the cell type, all the non-neurons, and you can also explore all the GABAergic neurons, as well as the glutamatergic neurons. And you can also explore more detailed structure for the subtypes, or for example, you are only interested in the astrocyte, you can explore the subtypes for astrocyte. And here, let me just show you one example I just talked about is the radioglia-like cells from two neurogenesis stone, as well as the neuronal progenitor cells. So this browser is very powerful because we built it using the UCSC cell browser. You can split the wheels, and then for one of you can still visualize the cell annotation. For the other view, you can, for example, check where the cells are coming from. For example, those cells came from hippocampus, and here are two populations of cells from two brain regions. And also you can check out your interested genes. For example, we know SOX2 is quite highly expressed in the radioglia-like cells from subgranular zone. And here I'm checking the open chromatin signals at the promoter region, as well as the gene body for every gene. But I need to make this clear to everyone. This is not means this gene is expressed. Here I'm only showing you the open chromatin signals for every gene. Okay, so this is the basically the function for cell browser. And you can explore other options. For example, you can also check out the detailed brain regions for every single cell. For the cell type, we offered a table, and you can basically search your interested cell type. For example, you can search for the cell types in the oligodendrocyte maturation, or you can also search for the radioglias if you are interested in. We also offer some detailed information, so including, let me show you one example. Like we offer how we did the QC and where the cells come from. But I guess you don't have to go to this detail, I guess. Another function I really want to show you is the genome browser wheel. We create a tab here called tracks. From this page, basically you can explore the open chromatin signals in every cell type. And this is built using the IGV. You can for sure zoom in, zoom out to see the open chromatin signals, as well as to changing the name of this track, changing the height, color, and also changing the data range. One thing I meant to mention is we offer not only the open chromatin signals, but also we offer the candidate cis element or the peaks we find from this study. And as I previously mentioned, we try our best to link every candidate cis element to its potential target gene. So this is another information you may be interested to look into more deeper. And to remove the cell type or add the cell type you are interested, you will need to click this tab called the setting, and you can uncheck the cell types you are not interested, and you can add another cell types you're interested. So the browser will update. The last thing I want to mention is we also offering a lot of results we already spent a lot of time and resource to get. For example, you can download the signal tracks for each cell type. You can download the arrays or fragments in every cell type. And you can also downloading the count matrix for every sample. You can also download the candidate cis element, a list of candidates cis element we find from this study. And for sure, you can also identify the links we find between a candidate cis element to its potential target gene. If you're interested in detail, do check the scripts and code we use for this analysis. And there are also several useful tables to provide more detailed information. You can find them here. And if you have any questions about the current data set or you are interested in some cell types, we can definitely help you to find it. And I think we are right now busy with analyzing the other half of the brain. So we are hopefully to also update this data set to the same proto later. So feel free to contact me or the other co-authors. After that, I have one last slide to show you. I know most of the people here must be interested in the brain, no matter it's a human or mouse brain. But within the same framework, the same structure, we offering other data set for you to explore. As I said, this is the mouse brain data set we have. I use this as a demo. And you can also explore the histone modification data from the mouse cortex. So we developed one single cell multi-omic technology called a pair tag. So basically this technique will profile the expression level as well as histone modifications at the same time. So if you are interested to check the histone state for every candidate cis element, feel free to check out this link. And we also provide another resource is where when we are more focused on identify the cell types and candidate cis element in different human tissues. If you're interested in the cell types in other tissues, do check out this data set. And there's another data set we are more focusing on the human heart. I also provide the links here. I think I will share the presentation I gave for today and to somewhere you can easily find the links. But also feel free to you can feel free to email me for more information.